most of this talk was built with Joel Meffert, who's a graduate student in my lab. The research that comes at the end is mostly his, and he's finishing up this year, so a lot of the people that he respects are in the room. If you are, if you're hiring postdocs in your lab and you like this stuff, well, if I do a good job, it's because of him. If I do a bad job, he's still really great. And you should talk to him about postdocs. So the tutorial's on linear mixed models and genetic association studies. So just as background, we're doing a genetic association study. So we have some phenotype that we're interested in. We collect DNA from some individuals. We look at some SNPs and we genotype them, and we get this matrix G, which is our genotype matrix. Gij is the genotype of the ith individual at SNP J. Now, we want to figure out which SNPs are associated with our phenotype. More specifically, we want to know which SNPs are either causal to Y or are in LD with one of the causal variants. And so we can look at our genotypes and look at our phenotypes and try to figure out whether these things are associated. But to do this well, we're going to assume a model. We're going to take the same model that was in the last talk, which is this additive model. So we have Y is G beta plus epsilon. Epsilon is going to be normally distributed. If we want to think about one individual, it's this sum, and the betas are the effect sizes. Okay, and if we do this, then we end up with this likelihood, just the normal multiple linear regression formula. And to find these betas, to maximize the likelihood, this is equivalent to this kind of optimization. Or if we want to think about it graphically, it's finding the vertical distances from the points down to the line. All right, and conveniently, we can do this analytically with G transpose G inverse G transpose Y. All right, so the first problem that we encounter when we're trying to do a genetic association study is that the number of SNPs that we have is much larger than the number of individuals. And so what do? Well, we say we're going to accept some type 2 error, and we'll just test each SNP marginally. And so all of the other SNPs that aren't this GJ SNP get pushed into this error. And now we estimate beta G this way. And so there's lots of different score tests or test statistics we can compute. I'm going to talk about this one, the correlation between G and Y squared times the sample size. And so we could also do LRT wall test or score test, but this is going to help us think about linear mixed models. So if we have this correlation between G and Y, and we let Y prime and ZJ be the centered and scaled versions of Y and Z, we can rewrite the correlation this way. And so our chi-squared test statistics looks like this: Z transpose ZJ inverse, ZJ transpose Y squared. And so just remember that. I'm going to see it again in sort of a different form in a few minutes, but it's this correlation between Y with some kind of normalization for X, all right? So the second problem that we encounter when we're doing genetic association studies is this population structure problem. So if we have different populations and the populations have a different phenotypic mean, then any SNP that has a different frequency in the different populations is going to appear to be associated with our phenotype, even though it's not causally related to the phenotype at all. And we can solve that problem by including P in our regression, but we don't necessarily have access to P. So people have sort of used principal component analysis to estimate these different populations, and then we can include these in fixed effects in our regression. As an aside, if P, so here P is changing the mean of Y. If P changes the variance of Y, that's sort of a different problem, and something kind of interesting happens if you make QQ plots where we want our test statistics to fall along the diagonal, and we think about SNPs where in population 1 they're greater than the frequency in population 2, SNPs where the frequency is the same, and SNPs where the frequency is opposite, then if P has an effect on the variance, this will be inflated, this will look good, and this will be deflated. But when we make a QQ plot for the entire population, it looks really good under the null because this and this kind of cancel each other out. So this is sort of interesting is that if the reason for the relationship between P and Y is actually due to genetics, then you necessarily have a relationship between P and the variance of Y. So if there's been selection that's causing this difference in mean between populations, you're sort of in this situation. And if you want to hear more about that, Shaila Musharraf, who's a postdoc in my lab, is here, and I think would be really happy to talk with you about that. Yeah?
Can you say a little about how, I mean, to me it doesn't, I don't think about these sort of things usually, but it doesn't strike me as that plausible a priori that the population structure would lead to a shift in mean. It seems like it would lead to sort of like segregated, like you have different subpopulations with different betas, like you want some kind of mixture model or something like that.
So most of the time the betas tend to be the same between populations. So if you have it at a causal variant, if this SNP causes you to increase your height by 2 centimeters in population 1, tends to have the exact same effect in the other population. But what can happen is the frequency of that SNP can increase. So if it's a rare height increasing SNP in population 1, but a common height increasing SNP in population 2, then just thinking about that one SNP, the other population will be a little bit taller. But maybe a bigger effect, probably a bigger effect, is that if you think about the environment in one part of the world versus the environment and the diet in the other part of the world, you can have large effects on phenotypic means that have nothing to do with genetics, but will still cause biases in your GWAS because you're not accounting for the difference in mean. So this third is, yeah.
Is it enough that the environmental variance differs between two populations to cause this latter issue?
Yeah, you don't need it to be, if the environmental variance, you'll still have this exact same problem. All you need is that the variance is different between population 1 and population 2. And if that exists, you can, in addition to sort of fixing this bias, you can squeeze a little more power out of your association study too. So another type of structure, some people think about these things as the same thing. I think about them as different, is related individuals. So if you have related individuals, like you have a family in your GWAS, then this assumption about all of your errors being IID isn't a good assumption anymore, because you have families share diet, they share their mealtime together, right? They watch TV, whatever it is, and that can affect your phenotype. And so maybe we want our phenotype to have this sort of correlation structure now where F is showing us family membership. And so if we compute this statistic and we don't account for this sort of correlation of errors, we're going to get inflated test statistics and we have bias again. And so we want to solve all three of these problems. And so one of the solutions that's become quite popular and is widely used is something called a linear mixed model. And so in a linear mixed model, and particularly this specification of a linear mixed model, which we use in genetics, we have Y again with, now we're thinking about these SNPs marginally, right? And we have some sigma, which is going to be K sigma G squared, where I'm going to tell you about K in a second, plus I sigma X. So this is the identity, right? So you have some covariance of the errors. For sort of mathematical convenience later on, I'm going to find delta as the ratio of these two things. And we can write down our likelihoods, which don't look as nice as the marginal likelihood, but we have Y with some mean and some variance. And if we take the log, we get this type of thing. And so the score test, if we want to adopt this model for how we're modeling our phenotype, looks a lot like the score test that I wrote down before for just the regular association study at marginal SNPs. So we have this G times G here, and we have this G times Y. So it's like we're looking at the correlation between G and Y as we were when we were doing just regression, except we're sort of twisting our Y a little bit by this sigma inverse. And we're accounting also for this noise structure in the genotypes. And I'll talk a little bit about exactly what that's doing later, but this is our score test here. And so when we want to run a linear mixed model, there's sort of three things we need to think about that are different than what we were doing in our linear regression. So first of all, these sigma g and sigma e, there's no convenient analytical solution anymore to find these optimal parameters. We have to choose what value of k we're going to use, how are we going to build k, and how are we going to define k. And we need some way to do our hypothesis testing, and even though I wrote one down here, maybe there's some other types of tests we want to consider. Okay, so let's talk about this problem one, this polygenicity problem, this m greater than n problem first, and how we might choose k to sort of regain some of that loss of power. So if we now have maybe a more complete definition of y, where we add in some fixed effects here, and we have the SNP that we're interested in, now we're going to look at this z, where z is just the centered and scaled genotypes. But now instead of having these betas be fixed, I'm going to make them random variables. All right, if we write down this model, we've now written down actually a phenotypic model, and the phenotypic model that we've written down is called the infinitesimal model. And that model says that all of the SNPs have some effect, or that is, the effect size variances are distributed like this, and that the rarer the variant is, the larger the effect size. And if you don't like that, maybe we can change some things later on, but let's just start here and see what happens. So if we accept this distribution of the phenotype and we think about this term ZB, which is supposed to capture all of the polygenicity, we can ask about what is the variance of ZB here. And it turns out to be this, because of how we defined beta. It's just sigma g squared ZZ transpose over m. And so we end up with a natural definition of k, which is the one that we use. So this definition of k is sort of the right definition of k to use if we want to account for polygenicity when we're running a linear mixed model. Later, I'm going to show that this sigma inverse term in our score test is proportional to the residuals of y minus a prediction from this ZB. So in effect, what we're kind of doing is looking at the correlation between the genotype that we're testing and some kind of, in our phenotype, but having subtracted out a prediction from this ZB term. And that's how we're winning, that's how we're getting power back by using a linear mixed model. Okay, so for relatedness, this k is also kind of a nice choice. So if we have these families, and we think about kij inside of a family, the kij of two individuals is some measure of their genetic relatedness. And so for siblings, kig is going to be about 0.5. And for each of those different relatedness structures, that k is going to correspond to their relatedness. So the assumption here is that the environmental correlation of related individuals sort of falls off as a function of their overall genetic similarity. So siblings will have an environmental correlation of 0.5 times sigma g squared. Avuncular will be 0.25, and kind of so on. So for population structure, what you can do, oh man, there's two yellow boxes here. Yeah, just squint. So if you simulate two populations, like John did in his talk, and you plot this matrix K, you get these sort of two blocks. So you have environmental covariance between these populations. And so it's kind of getting at this population structure too, but that matrix K that we built, that definition that we used for K, is the same definition that's used when we're doing principal component analysis. There's kind of this neat parallel. And later, if there's time, we can show that this linear mixed model is basically, or is ridge regression over all of the PCs. And if you want to sort of understand more about why this is a good choice and how it relates to what you can and can't do with population structure, this is a nice paper that kind of goes through a lot of the theory. Okay, so now that we've chosen our K, as this, which is going to cost us quite dearly of n squared m to build this k. We'll talk about estimating the parameters of our linear mixed model. So we've got the beta for the fixed effects, the beta for the genotype, and then these two variance component terms. Yeah.
So if you want to correct for all of those, do we have different k's or just one k is sufficient?
So if you just use 1k and you make family structure and population structure, then you end up with some bias. And so you need to add in, so it's not like magic. It doesn't get everything all at once because you've sort of misspecified what that sigma g should be. Unless you get very lucky in how you design your population structure and relatedness, it's not getting the right sigma g for both of these things all at the same time. And so you might want to have multiple sigma g's to account for different pieces of this. Gian has a paper, in Perspectives paper in Nature Genetics, I'll give the citation later, that sort of discusses this issue. So the way that you find these sigma g, the first approach that was taken was like a maximum likelihood approach. So we can write down, before I get there, there's this sort of parallel between unbiased estimation in linear regression and unbiased estimation in this linear mixed model context that we have to kind of talk about first. So there's this thing that you'll see when you start reading about linear mixed models called the restricted maximum likelihood. And so when we have these other fixed effects, so W now is going to be all of our fixed effects, X and G, and we can write down something called the restricted maximum likelihood, which has the original likelihood plus some extra terms. And where does that come from? Well, here's the sort of parallel. If we do regression and we have P sort of fixed effects, the way we get an unbiased estimate is to divide by n minus P instead of dividing by n. And so similarly, by adding this sort of constraint to our regression, we're getting an unbiased estimate of the sigma g by doing the restricted maximum likelihood. And if we just do a maximum likelihood, we are not getting unbiased estimates of our parameters anymore. So to maximize, we can try something like a grid search. And this is going to be very painful because within our likelihood, we have these inverses. And so this matrix is n by n, and so the cost of an inverse is n cubed. And so if we did this, we have to pay a lot of these inverses. So maybe we'll do something a little bit smarter. So we can try this gradient descent. So we can do something just like this standard, take a derivative, move down, get a new estimate. What's done in practice is to use a second derivative to move there faster, so we pay even less of these matrix inversion penalties. And so if you see this AI REML coming up, it's this kind of way to take an average of two different types of estimates of the second derivative. And this is what's implemented in a lot of different packages for doing linear mixed models, like, for example, GCTA. So for each choice of sigma g squared, we're still paying this matrix inversion, and so our total cost is the cost of the construction of k and the number of matrix inversions that we have to pay in the estimation. So this is hard, it's expensive, especially when the matrix is big, and so there was a lot of work done to try to speed this up. And one of the first approaches was to think a little bit more about this matrix K. And so for a matrix K, you can do a spectral decomposition. You can represent K as this U lambda U transpose, where these are the eigenvalues and these are the eigenvectors. And so how does this relate? Well, if this is our sigma matrix, we can rewrite it this way. And if we want to compute the inverse of sigma, kind of algorithmically, this is much more pleasant because we can just flip all of the eigenvalues and then remultiply by U transpose, and we get the inverse. Computationally, we've won nothing because you still have to multiply these two matrices together. But if you do a bunch of algebra, these two terms in the likelihood can be re-represented as elements of the spectral decomposition of two matrices, one that relates to this term and one that relates to this term. And you can read about that in this paper. And so you still pay O of n cubed for every SNP, but you only pay it once. You don't have to do any more searching because the search for the optimization is now linear because we've done this spectral decomposition. So we've won a little bit. The next kind of big advance came sort of at the same time from the Microsoft group and from the Stevens group. And they did it sort of differently. This one is a little bit easier to talk about. And so the idea here was to take this U and just twist the phenotypes and twist the fixed effects. And so what that does is you get the same likelihood, but the sigma is now diagonal. And so for every choice of sigma g, you just pay a linear time to evaluate the likelihood. And so you can very quickly, so you pay one spectral decomposition for the whole k, and now every time you want to choose sigma g, you can just quickly compute the likelihood. The other advance that's described in the same paper is if you have a situation where m is actually less than n. And so what they do, so here's our spectral decomposition, and here's a singular value decomposition. And so this is the same u, and this s squared is lambda here. And so if you do this, you can actually never build k, which is nice because it was expensive to build k, and you can get back u and lambda. And so if you don't want to do a multiple linear regression for some reason, you want to run a linear mixed model, you're now only paying this cost, and you're not paying the cost of the matrix construction. So these are sort of the set of exact algorithms for the exact tests, and all of these sort of speed ups. AI-REML, you can have multiple Ks, but these sort of speed up tricks, the MX trick and the fast LMM and the Gemma trick, you can only have one K. So far we've been paying these, we've had to re-estimate sigma g to do it properly. You have to re-estimate sigma g squared for every single SNP that you're testing, which is very expensive. If you're willing to assume that the beta g is quite small, then these are going to be approximately the same for every single SNP. So we can estimate sigma g het squared once, and so we can really speed everything up, but this only works if the effect sizes of the SNPs that we're testing are very small, which is true in genetics and in practice. And so this is a very commonly used trick where we estimate sigma g squared once under a null, and then we just test every SNP with that same sigma g squared. So the last, it's too bad Poru left, this is a really cool paper that he wrote, is that instead of spectral decomposition or this AI-REML, we're going to do some Monte Carlo sampling to get to sigma g squared. And so he used the results of this paper, which said we can compute the sigma inverse x using this preconditioned conjugate gradient descent. And so if you can compute this really quickly, you can sample from the distribution of y conditional on your choice of sigma g and sigma epsilon squared. And so we can sample from the space of the parameters of the model until we get to sigma g squared. And so this is O of mn, and the number of times in practice that we have to sample is n to the 0.5. So now we have described the three different approaches, this gradient descent, the spectral decomposition, the Monte Carlo sampling. And I'm going to talk a little bit about hypothesis testing because this is also an expensive part of the procedure. So this is the score test that I wrote down. And so this numerator, again, is this sort of correlation between G and this prediction of Y. So the other thing you might think about is that if you have So first, this is where this comes from. So there's the prediction. So we've made an estimate of the distribution of y, so we should be able to predict it. So under a linear mixed model, this is the predictor for the phenotype. And so if we think about y minus the prediction, the residuals, it looks like this. And so it's sort of our sigma inverse y. Huh? And so it might occur to you, well, we should just take these out and do the regression, and this is done in this paper that was called Grammar, and it's really fast because I don't have to. There's no inverse here. I just make one residual and I just do regression over and over again, but it's biased because it ignores this denominator. Or in other words, this predictor is overfit. So there were two papers that have relied on sort of an adjustment to account for this bias. The first is this Grammar gamma, and so they still are doing this regression, but they try to fit sort of an average estimate of the error of what the denominator should be per SNP to get rid of the bias. Bolt also uses this regression of genotype against y minus y predicted, and it rescales its genotypes through LD score regression, but they're both relying on this residual analysis. So that kind of brings us up to speed on the state of the art of methods for running linear mixed models for genetic association study. So now I want to talk a little bit about some extensions and issues, and then tell you a bit about what Joel's been working on. So it's a rich field. So there's a lot of issues with these things. One is that if you're running in case control data, when you ascertain data, all of the things that are causal to y become correlated, and so you end up with biased estimates, and you end up losing power. And so there's some methods that discuss that. The link function that we used is this linear link function, but you might want a negative binomial, or you might want a Poisson, or you might want a logit. Note that if you think, oh, I'm in case control data, I'm going to use the logit, that the Prentice and Pike result doesn't hold anymore for a linear mixed model. So you can still do logistic regression, but you don't have this wonderful guarantee about the odds ratios being consistent regardless of ascertainment. This is something that Aheki brought up. There's this multi-BLEP approach to allow for multiple variance components at once that has some really nice mathematics to make the computation more efficient. And then this is Bogdan's question, basically. If you want to know about running everything all at once, you can check out this paper and see where it breaks and where it doesn't. If you were here for CGSI, Sriram talked about this mailman algorithm for fast matrix vector multiplication. Bolt uses this, and so in principle, you should be able to speed up Bolt even further by implementing this mailman algorithm for the matrix vector multiplication. So, what time did I start? I have 15 minutes? Thanks. All right. So this residual analysis that we did, which was that these other methods used, was this residual after predicting y. But you had a sample event and you were fitting m things. You're gonna overfit. You're also fitting in sample, and so this the correlation, because you've overfit between the genotype or the noise term in your model, is going to be correlated to your predictor. So that's kind of why you're getting this deflation in a sense. And so what you might want to do is, instead of using the predicted y in your sample, is you might want to build your predictor or estimate your sigma g and sigma epsilon in n-1 individuals, and then predict into the nth, and sort of do that over and over again. And if you do that, you get a cross-validated prediction of y, and it's not correlated with the error anymore. So you've kind of solved this problem. It's just very expensive, because you have to run this linear mixed model n times. And so, what Joel kind of worked out based on some theory that's probably familiar to a lot of you in the kind of machine learning side is that you can estimate the out-of-sample errors as long as your predictor is of this form y, hy, okay? And so with some algebra, you can show that the out-of-sample BLUP, the out-of-sample prediction of I, comes naturally to you once you've estimated your linear mixed model. And so in linear time, you get out-of-sample predictions of all of your out of all of your individuals. So if you're running MX or you're running GCTA or whatever it is, you can add, a couple lines of code and get the out-of-sample predictions of all of the individuals. And so let's talk about these CV BLUPs a little bit because they're kind of cool. I'm going to save the sort of bit about association testing for the end because it's kind of the obvious thing, and there's some other stuff that Joel and some other people in the lab have been doing with these cross-validated predictors that I think is maybe more interesting to talk about first. So first, I want to tell you about this relationship between principal components. Oh, I have the wrong title here. So anyway, you have, if you remember our principal component, our spectral decomposition of K gave us U lambda U transpose. And we sort of had this problem when we're doing this regression, which John talked about a little bit yesterday, just how many PCs do I want to include? And choosing the top isn't such a great thing because you can have stuff in the middle that's correlated with the outcome, and it's a sort of challenging problem. And so what you might think to do is do a ridge regression over all of your PCs. So if you write that down, here's a sort of transformation of our PCs by lambda, so that in our penalization we're sort of upweighting the PCs that correspond to the larger eigenvalues. So we can now consider this way to estimate the amount of weight we want to put on each PC. So we end up with a predictor of y from the PCs that looks like this, where if we, lambda is our penalization term, but if we specify that lambda is related to the parameters that we estimate or that exist in heritability space, our predictor is exactly our BLUP. And so PCA is ridge regression over the PCs is BLUP, and if we do this cross-validated, then we've done out-of-sample, basically ridge regression over all of the PCs. So it's a way for you to effectively choose which PCs you want to put in your model when you're running a linear mixed model through ridge regression. So another nice thing that comes out of having these CV BLUPs is that you can, Yeah.
So that's kind of a natural weighting that you're assigning.
Yeah.
But in practice, we could actually decide to change that.
You could weight them differently, yeah. So another way to think about linear mixed models is as Bayesian regression. We have some prior on the effect sizes. And if you think about it that way, because you're now doing this penalization, you get a shrink of every single beta. And that shrink is a function of the number of causal SNPs that you have. And this is sort of an interesting thing that you might want to be able to pull out of your study. There's a lot of reasons you might want this shrink. It has a lot of different applications. But one that's sort of simple to understand is that it's related to the fraction of causal variance as long as you know the number of independent samples in your study. And so if you do a prediction, the variance of that prediction should be sigma g. And if you look at the variance of your predictor, it's shrunk. It's not sigma g because there was this shrink that happened. But if you can look at the variance, you can show that if you look at the variance of the out-of-sample predictions, its relationship to sigma g is exactly that shrink. And so if we do a simulation where we choose some fraction of SNPs that are causal, we have some theoretical shrink. If we look at the variance of these BLUPs related to sigma g squared, we get this sort of poor estimate of the shrink because we're doing it in sample. But if we take our out-of-sample variance of these CV BLUPs and we relate that back to sigma g squared, we get a good estimate of what the shrink was. And so in particular, if we know the number of individuals in our sample, we can tell you something about the number of causal variants that are in the phenotype. So this shrink is doing something really nice for us in terms of genetic architecture of the phenotype, learning about the genetic architecture of the phenotype. Yeah.
So will the number of SNPs in LD with your causal SNPs affect the shrink?
Yeah, so the shrink works with LD, but this is the number of causals is something a little strange when you have LD. And so the shrink is going to be proportional to the number of causals, but it's also going to be an issue of where those causals are living with respect to how many LD pairs they have. And so it's telling you something interesting about the number of causals, so it's telling you something about the genetic architecture, but I think it would be quite a stretch without making some heavy assumptions. Maybe you want to assume that SNPs are distributed randomly, and then you could do it. Estimate actually the number of causals. So let me say that actually clearer. If you were willing to assume that the SNPs were distributed randomly, you could estimate the number of causal variants. But it's unlikely that causal variants are distributed randomly, and so you're going to be wrong.
How far from how wrong?
I think.
The estimate be of any.
So, right, yeah, the estimates, yeah, sorry, that was kind of a negative note. It's still interesting because comparing the shrink between different phenotypes will tell you something about the different number of causals between those phenotypes. I think a maybe more reasonable assumption is that however non-random they are, their non-randomishness is shared between different phenotypes in a similar way. I'm not saying that they all have exactly the same sort of LD properties, but it's going to be much more similar than it is to random. So if we have lots of phenotypes and we look at the shrink across lots of different phenotypes, I think using that shrink to learn about which one has more or less causal variance is a reasonable thing to do.
Does it make
Have you
sense?
tried this?
Yeah.
Again, height versus
Yeah,
some other
yeah,
trait?
yes, yeah. Stay tuned for the preprint. Okay.
Would it make also sense to look at different chromosomes and where N is actually the same?
Oh, that's a really cool idea. Yeah, that totally makes sense. Yeah, absolutely, that makes sense. Yeah, we didn't think about that.
That ratio, right?
No, yeah, so you could sort of learn about different, like, yeah, that's really cool. We should do that. So another thing you can do with these cross-validated SNPs is cross-validated BLUPs has to do with testing for epistatic interaction. So we've been thinking about this sort of omnigenic thing or this polygenic buffer thing or however you want to say it, but here's sort of the rough model, which is that we have some set of core genes, we have some set of general genes, buffer genes that are talking about general cellular health, and if this phenotype here sort of modifies the effect of those core genes on some phenotype. Okay, so it's a model. It may or may not be right, but it's a model. Okay, and so if we want to believe in that model or test under that model, you could say, I'm going to make a CV BLUP in sample from these core genes. I'm going to make a CV BLUP for these buffer genes, and then I'm going to test for this interaction. And so I've got these two predictors from gene set 1 and gene set 2, and I'm asking whether the phenotypic prediction from gene set 1 modifies the phenotypic prediction from gene set 2. And if we want to write down a model like that, you can kind of guess at these core and buffer, but you're probably going to do a bad job because we don't know what every gene does. But it turns out that if you just divide the genome into odd and even, and this is the true model, you're still going to have quite some power to detect the effect if this is the true model. And the other nice thing that it does for you if you do odd and even chromosomes is those chromosomes are no longer in LD with one another. So you don't have to worry about this interaction effect showing up just because of LD that's independent of population structure is still an issue. I want to highlight, I put this in here because Poru did this for me last night, so thank you, CGWI, for making things happen, right? So he took the UK Biobank, he took all of the unrelated European, British population, split it in half, ran Bolt LMM, got a BLUP predictor, and then made a predictor for the odd chromosomes, made a predictor for the even chromosomes, and made an interaction term. And the p-value of the interaction between those two things was quite significant. We did it previously in NFBC, which is a much smaller cohort, and we didn't have type 2 diabetes, we had fasting glucose, but it was significant. Other phenotypes were not significant, like height and BMI, and so it was pretty interesting. Yeah?
So the type 2 diabetes will on the observed scale or the line?
Observed scale.
Or that could be-
It can, the reason UK Biobank was kind of nice is because it's not ascertained. But you're right, if it's ascertained data, this will also be biased. But you're right for two reasons. There's also this scaling issue. An interaction can be non-existent on one scale and existent on another scale. And so, this may or may not, in the case of type 2 diabetes, be a statement about biology because of the scaling issue. But what it is definitely a statement of, provided it's not some sort of confounder from assortative mating or population structure, is that if you're trying to do prediction, you can do a better job of prediction by including the interaction term. Because if you're trying to predict on the same observed scale. So it might be that really there's no biology here. It's just a sort of scaling thing and that we're on the wrong scale. But if you're trying to predict on the observed scale, I just said that. I'll say it again. You'll do a better job with the interaction term than without it. So this is related to the last talk, where you're trying to predict gene expression, but instead of using it to predict a phenotype out of sample, you could use it in sample to predict the expression of other genes. And so if you are trying to do trans-QTL association studies, you can use the CV BLUPs at gene A to predict the expression at gene B. And so this gives you a really powerful and fast way to test for trans effects and also cis-trans interaction effects. So I can look at the effect, the CV BLUP of gene A, plus the CV BLUP of gene B, plus their product, and see whether that impacts the value of either gene A or gene B. And so if you do this, you can find many more trends than you would through this, also much faster. And the other advantage is that these programs, SVA and PEER, are sort of meant to be run conditionally on the variables that you're testing. And if you just chuck them in, you can get bias and power. But if and there's no way in a trans-QTL study you can run SVA for every single SNP phenotype pair. But when you're doing CV BLUPs, you can run SVA conditional on each gene, and then you can test using those conditional SVs, those conditional PEER factors. So it's kind of a nice feature of doing trans-QTL studies this way, is that you avoid some of the bias that can creep into trans-QTL association studies due to these noise factors that are shared across lots of genes. The CV BLUP, the correlation of the CV BLUP is proportional to the genetic correlation. So if you run lots of phenotypes, like in the METSIM, you can very quickly estimate the genetic correlation of many hundreds of phenotypes at once. And I'm going to skip the association testing a bit because I think I'm way over. Sorry. And just thanks to all the people who were involved in the Free Software Foundation. Thanks.